a PhD in computer science from the University of Maryland, College Park in 2010. He was then a postdoc in the machine learning department at Carnegie Mellon University before starting his lab at the Salk Institute in 2014. His research interests include designing algorithms to study the structure and function of biological networks and the study of algorithms in nature. In 2008, he was named a Pew Biomedical Scholar. Saket's talk today is titled Network Design Principles of Plant Shoot Architectures. Let's welcome Saket.
At the symposium, it's honestly a little bit of a new world for me, coming from computer science, but I'm hoping we can connect in some new ways here. So what I want to tell you today is what the role of theory could be in terms of plant biology and phenomics. And what I want to tell you about is a theory that is fundamentally about trade-offs in biological systems. Okay, so imagine you're trying to build the beak of a bird. Okay, you could build this beak to be great for picking up worms out of the ground. You can also build a beak that's really great for flight and aerodynamics. Okay, but both of these tasks are important for the survival of the species, but clearly these are not the same beak. And in fact, performing well on one of these tasks may actually inhibit your performance on the other. Okay, so this is a problem that evolution has to solve, and the idea is that if both of these are really important for survival, then evolution might have come up with some good compromise or some good trade-off between these two things. Okay, but the question is how do we know that? And how do we actually go in and quantify how well evolution actually did in resolving these kind of trade-offs? So I'm not going to talk about beaks, but what I want to tell you about is about plant architectures, and I want to tell you about how we actually got into this problem from the background that I came in from engineering. And the motivating example that we started with is, let's say that you wanted to build a transportation network, okay? You wanted to build a subway system that connected people out here in the suburbs who all want to get to downtown. They want to get to work quickly, okay? And your job as an engineer, as a network design engineer, is to really just build the network that connects all these nodes up together. Okay, and so the question is, how would you design the system? And usually in engineering, the first thing we think about is, okay, what is the objective of the system? And then we can design to meet that objective. Okay, so here's one reasonable objective that you might want this kind of system to have. Okay, you want to minimize travel distance, right? People want to get from home to work as quickly as possible. Okay, and then you can ask, right, if this were my only objective, minimize travel distance, what would be the optimal network that would solve this problem? Okay, and it's very easy, right? Everybody just beelines it straight in. Okay, everybody's very happy. They get there as quickly as possible. So what is the problem with this? The problem with this is that it's very expensive to build, right? That's a lot of rail you have to put down. It's a lot of money. It's a lot of manpower. And while this is a very well performing system from the user's perspective, from the builder's perspective, this is a bit of a nightmare. So here's another objective, just like in the bird case, that introduces some tension between this guy. And that is, let's say you wanted to minimize your cost, or literally the total amount of rail you actually have to put down to connect everything up. Okay, and then you can ask a similar question. What would be the optimal network that just minimizes the total length and gives everybody at least some way to get to work? And so this is another network. It's called a Steiner tree in computer science. And the idea is that it's a network of minimal length that allows everybody to get to, even though, you know, this guy, for example, gets a bit screwed in the morning. He has to go all the way around. Okay, but you're given and you have these additional points, these Steiner points or branch points that you can use to kind of minimize how much rail you need. Okay, so what we have here are two networks that optimize different objectives. So this, let's just call this guy the satellite. He kind of looks like a satellite. Great for minimizing your travel distance, terrible for cost. Here's another guy who's really cheap to build, but you know, there's a lot of people who are unhappy. Okay, and the question is, how do you resolve this tension between these two things that are, you know, working on each other? Okay, so what does this have to do with plants? So the analogy that we made was let's say that instead of this downtown, you think of this as the base of the plant where the root system is emerging. And you think of the suburbs as the locations of the leaves of the plant in three-dimensional space. So what does minimizing travel distance mean to the plant? It's a measure of how efficiently nutrients can be transported up and down from the roots to the shoots. And so, for example, sugars that are generated by the leaves due to photosynthesis and traveling down in the root system, and nutrients that the root system is collecting and back up. Okay, so this would, in theory at least, be the architecture that a plant would want to use if all it cared about was minimizing its transport efficiency. And on the other hand, you can ask the same question about minimizing total length, and that has to do with how many resources are actually needed to build this architecture. So for example, the amount of carbon you need to actually build all the branches that you need to connect these leaves and where they need to go. Okay, so are these the right objectives for thinking about plants? I don't know, but let's just hang in there for a second and just think about, let's say these are important in some way, or they're correlated with something that's important. How well would a plant resolve this tension between these two objectives? Okay, this is what I want to talk about today. And so I just want to just start by formalizing this problem a little bit mathematically, and just to give you exact sense of how we're going to tackle this. So as input, I'm going to be given a set of endpoints. So these are going to be points in three-dimensional space where you tell me where these suburbs are or where these leaves are located. And a point P0 corresponding to the downtown, or where the root system, where the base of the plant is. And what I'm going to give you in return is this network. So this set of branches B. And our two objectives, just to formalize them, is to minimize the travel distance. So what I want to do is minimize the distance along the branches from that point P0, that point in downtown, to each one of these leaves. So just sum the distances from the base to each leaf. And my second objective is to minimize my total length. So this is just going to be the sum of all these little branches that I actually build to make this network. Okay, and just to kind of give you a sense of the phenotype space that exists with this characterization, even if I just had 19 points, so in other words, I just had 20 leaves or 19 leaves on this tree, there are 5.48 times 10 to the 21st possible networks that you could build that connect up all of these leaves together. Okay, so it's a huge design space that we're in, and now we just let's actually dig into this space and see what it looks like with respect to these objectives. Okay, so here's my design space, my phenotype space here, and I'm going to evaluate each of these phenotypes according to the two objectives that I talked about: the total length and the travel distance. Okay, so what do we know about this space? So the first thing we can do is put our two guys who we started with. So this was that Steiner tree, the one that has the minimal length. So it's great in terms of minimizing length. It's out here on the left, but pretty bad in terms of the travel distance. Okay. The other guy we have is this satellite, the guy who looks like a fan. And so this guy is great in terms of travel distance, but very expensive to build. Okay, so what else do we know? We know that you can't do better than this guy and better than the satellite in terms of travel distance. So you cannot possibly be down here. And likewise, you cannot possibly be left of the Steiner in terms of the cost. Okay, and so what we really hypothesized, this is all, again, this is all starting from theory. What we hypothesized was that if these objectives are in some way related to plant fitness or important to plant fitness, then there might be evolutionary pressure to try and drive down these costs, to try and get as close to maybe this magical point as possible, even though maybe that's not even feasible. And this seemed like a reasonable thing to test for plants, because A, there's a lot of genetic diversity, and B, they've been around for millions and millions of years. So if evolution has all this time and diversity, you might expect that it would find a good way to get, as low as you can possibly go. But what is the best you can do, right? It's not clear, like, can I literally get to this point? Is there a network that would get there, or is that the best point, or is that the best point? Or, there's no way that we have currently to characterize how do you balance between these two different objectives. And the idea is that, you know, maybe I care about 80% of this guy and 20% of this guy, or maybe I care 50-50. And what we need is some way to kind of combine these two into a single objective and then look at all the different ways to interpolate in between them. Okay, so that's what we sought to do. We wanted to come up with a way to combine these two, and we literally did the simplest thing possible. So we took our two objectives and we just came up with a linear interpolation in between. Okay, so in other words, instead of just minimizing this or just minimizing this, I'm going to minimize some value alpha times this objective plus 1 minus alpha times that objective. Okay, and you can think of alpha as literally just a weighing term. That's saying how much do I care about one versus how much do I care about number two. Okay, and so just by looking at this, you can see that if alpha equals 0, then this guy goes away, and the only thing I care about is the second objective, and the best thing to do is the Steiner tree. If alpha equals 1, then this guy goes away, and this guy's left. All I care about is travel distance. And for a value of alpha in between 0 and 1, that's where I start to care about 50-50 or 60-40 or whatever you want to do. And for that, we came up with an algorithm. And I don't want to go through the details of the algorithm, but I'll show you what the result of this algorithm is. But what the algorithm does is it finds the network amongst that huge space that I showed you that very nearly minimizes this objective for any value. Okay, okay, actually, I'll just show you very briefly. So it just kind of, in every step, it just finds the kind of edges that connects and minimizes this in every step. Okay, we could talk about it later if you want. But so here's what you kind of build using this. So I'll tell you about this data in a second, but in each one of these plots, it's just a 2D projection of where the leaves are and where this space is. So all the green points here are the locations of where the plant put the leaves. And this black dot here is where the base of the plant is. And so using this, we can compute the satellite very easily. Just everybody connects straight in. We can compute this Steiner tree very easily. Here's what the actual plant did. And again, I'll tell you about how we figured this out in a second. And then here are the result of the algorithm for different values of alpha. So if alpha equals 0.25, actually, Paul, there's actually something happened here. I'm just gonna check something real quick. I'm sorry about that. Okay, it's okay. Okay, so here's what the tree. So if we use our algorithm and apply alpha equals 0.25, we get something like this. If we do 50-50, we get something like this. 75-25, we get something like this. Okay, so these are different networks that minimize that objective for different values of this trade-off ratio. Okay, and so what we can do is by using this algorithm, what we can do is generate what's called a Pareto front. And the idea here is that this is the optimal for alpha equals 0, this is alpha equals 1, and then we use our algorithm to kind of interpolate what the best thing to do is in between these two endpoints. Okay, and so the idea evolutionarily is, let's say there's a plant that somehow falls over here in phenotype space, and then a new plant emerges that falls over here, where then just due to fitness, this guy will eventually be eliminated from the population, and the only plants that should remain are those that fall along this curve. Okay, and where you fall along the curve might depend. You know, maybe in one environment I care about this one objective more than the other, and maybe in a different environment that changes. But this is kind of the space over which we would hypothesize these plants to lie on. Okay. Okay, so this is all theory, right? And so we really wanted to go in and actually test this idea using actual data. And so what we did is we did a lot of 3D scanning of plant architectures. And so we called this company, it was very interesting, Faro. So they mostly build these 3D scanners for building airplane parts. So like Boeing uses them to do 3D printing of parts for their airplanes. And so we called them up and said, you know, can we use this to actually scan plants? And so they were like, we've never actually tried this, but they came by and it really really beautiful data. And I'll show you about it in a sec. But really, like any technology, there's some advantages and disadvantages to this. So one of the advantages is that we get very high quality. We get micron level resolution. This is non-invasive, so you don't have to touch the plant in any way to actually capture the material. And it's not taking pictures and doing like foreground, background subtraction and all those issues. Literally, you scan it with the laser and all you capture is the plant material without any background. So in terms of doing all the post-processing steps that are difficult for imaging, you don't really have to deal with those issues with scanning. The downside, it's definitely much less lower throughput. So that's Adam's arm right there, and he literally has to go and every day and scan plants manually. So that's going to be a bottleneck. And at least the scanner that we got has about a 6-foot arm to it. So this guy right here keeps you registered in 3D, and this you can move around and scan the plant. But if you're limited to a 6-foot arm, they have larger arms, but ours is 6 foot, so you can only scan over that size. But for our purposes, this was good enough. Okay, and so here's some data of what a tomato plant scan using this laser scanner looks like. So this is a tomato plant with about almost 1 million cloud points. This false color that's about to come up is false. It's just showing you the distance from every point to the base. You can easily capture leaf thickness. You can see leaf venation patterns in there, all the thickness of different elements. So it's very high quality data that we're still very untapped in terms of what is the other things that you can learn from this. But as I'll tell you, all of this data is available and anybody can use it. So that's what we did. So we thought, let's test this theory, our plants on this curve, by starting with tomato plants. And we grew them just in ambient growth conditions to start with. So that's what the scan would look like on day 4 with just the 2 cotyledons. And then you see the same plant on day 12. And then we stopped at about day 20 when it had about 20 leaves or so. And so we would scan the plants, and then literally what we would do, Adam would go in and literally just take points along the architecture. So he clicked on the base of the plant. We got some XYZ coordinates, and then we clicked on the base of every single leaf that emerged or leaflet, and we got those XYZ coordinates. And literally the only input to our algorithm was this set of XYZ coordinates that we used. Okay, and so then we tested the theory. Okay, and so I'm going to show you a bunch of plots that look like this now. So this is the same thing as I showed you before, with total length on the x-axis and the travel distance of the plant on the y-axis. And so this is tomato ambient conditions on day 4. And really, to our surprise, there the plant was. It was right there on the Pareto front, on that curve. So this was the Steiner tree, that was a satellite, and there the plant was. And we were like, wow, this is, what is the chance that this would have happened? And actually a reviewer asked us this. And so we said, okay, that's a very good question, actually. And so we said, okay, what if I just connected things up randomly? What is the likelihood that one of these guys would fall on or near this curve? And it was very unlikely. Most of them are way out here that I didn't even draw. So, and here's another architecture that, let's say all you cared about was building a structurally stable system. What would it look like? And this is also pretty far away from the Pareto front. So what this is telling us is that, again, are these the appropriate objectives? We don't know. But either they are, or they seem to be correlated with something that this plant might actually care about in terms of its growth and its adaptation. Okay, so fine, this was just one plant on day 4. What happens as the plant continues to grow? And man, this guy just kept hanging around there. So there he was, right on day 12. He was still hugging the Pareto front, and then even up to day 20, he was still right on the curve. Okay, and so we were, computer scientists, like, this is actually really amazing. What a system that evolved to do this thing. And so we went to the plant biologists, and they were like, okay, this is interesting, but basically all you showed was that a single species in a single very very easy growth condition abided by your theory. And they made a good point. I mean, like plants clearly have to grow and adapt to many different conditions, and there's a lot of plasticity in these conditions. And it's a good idea to test your theory because eventually it will break, but let's see how far you can push it before it starts to break. So we said, okay, let's try to repeat these experiments then in different growth conditions. So we took tomatoes and we exposed them to 4 other kinds of stresses. So we put them in shade. We droughted them. We didn't give them any water for 20 days. We put them in excessive light. We put them in excessive heat. And in all of these conditions, you can, I mean, just looking or just calculating basic features of the plants, you see, lots of changes. So for example, in shade, you see this classic shade avoidance response where the plant shoots up to try to get above the canopy. And so you can see its height has gone up by an order of magnitude compared to other conditions. And highlight many more leaves in other conditions, and the total volume, so this is the convex hull volume of the plant's cloud points, again changing by orders of magnitude. And so we thought, okay, so these are all different conditions, and does our theory still kind of hold to plants in this kind of diversity? And really it was still working out just okay. So this is shade, plant on day 13, right there on the Pareto front. Again, all these other things pretty far away. Day 21, it's growing. And you can see on the X and Y axis here, this plant has grown from, about 100 millimeters to, 200, 300, 400 millimeters. So it's really growing 2-3-fold, but still managing to constrain its growth process to lie on this curve. So this is in shade. We did it in drought. It's still right over there. We did it in high light. It's also right there hugging the Pareto front. And this is just a summary of all our data. So this is, on the x-axis is the day that we scanned the plant. On the y-axis, we came up with this distance of how far that plant lies to the curve. So lower here is better, expressed as a percentage of the plant's length. And so you can see all the actual data is very close to 0 and 1%, less than 10%, whereas these other baseline networks are much further away. So this was, quite striking to us to see that this species was able to do this. And so then we went back to the plant biologists again. And they're like, what do you think now? And they're like, okay, this is a little bit better. But, there's a million species of plants out there. Like you just showed tomato. Why don't you see, where else this thing could work? And so I don't want to keep showing you these diagrams, but we tried it in sorghum plants. We tried it in another dicot in tobacco. We did experiments over 20 days, and we saw a very similar story that these guys were also quite close in lying on this Pareto. So the other thing that was interesting was, okay, so they're lying on this curve, but is there any logic or is there any systematic order to where they fall on this curve? And so the idea here was that, so here we have this curve that we've shown, and what we found was that the location of the plant on this curve was actually an interesting measure of saying what kind of optimization the plant is trying to do. Okay, so I'll just give you an example here. So if we did this and we looked at where the tomato plants lie, they all lie somewhere around here. And then all the sorghums lie, closer to here. And this is maybe not so surprising because sorghums literally do look very much like this, as they are grasses. But actually the more interesting thing for us was what happens as we varied the conditions. So here are, if we put many different plants, they lie somewhere. This is just a schematic, but they lie somewhere here. And then you take that same species and you put them in high heat, and they systematically move over here. And this happens in tomato, it happens in sorghum, it happens in tobacco. And so what it seems to suggest to us is that there's some kind of feedback that's happening from the environment that's changing what kind of optimization this plant is actually trying to do. And it's happening in somewhat of a systematic manner that we could actually predict where that plant is going to go next. So so this was, some evidence that we showed that plants somehow evolved mechanisms to solve this very basic engineering problem, this network design problem. And there's a lot of things we're thinking about next, and I wanted to tell you about a few of these. So one is, again, from a computer science perspective, I'm fascinated that plants have come up with some algorithm to generate networks as they are. And this is a problem that's important in many areas of computer science. And so we're actually thinking about, what is actually the plant's algorithm for doing this, and how does this compare with other algorithms that are out there? You'll notice that what we did here was a very abstract notion of plant architecture. So we literally had skeletons and we were analyzing skeletons. There are many other features that we ignored. So for example, the thickness of branching elements, branch angles, leaf size. So all of these, of course, are clearly important to architectures, and it's something that we're thinking about of how to include some of this additional complexity into this model. But at the same time, we're somewhat pleased that you don't necessarily need all the complexity to find something that we think is interesting. So we're kind of balancing between what is the right level of abstraction here and how to deal with that. We want to know, so all of our experiments were done with single plants in isolation growing, but clearly competition is another factor that affects growth. And so we're kind of building now gladiator style arenas where we're putting two plants in the middle and having them battle it out and doing our scanning as these battle happens. So we're excited about that. And then I want to tell you about two things that in a little more detail is trying to get more at the genetics of how this might be happening, and then kind of stepping back and looking more broadly at where else this principle might be applying, both in roots, and then I'll talk a little bit about neurons as well. Okay, so the molecular basis. So the idea here is that, we found that there seems to be some systematic things that are happening that are shifting where these architectures lie depending on the condition, depending on the species. And so thinking about phenotyping and breeding, if we can control that, if we can breed a specific phenotypic trait in an environment or a species of interest, that might be a useful thing in terms of, you can tell me, but that sounds like it might be interesting in terms of breeding. And so what we wanted to do was see if we could actually identify genes that can be modified to kind of move or systematically move this architecture along the Pareto front. And to get at this, what we actually turned to was Arabidopsis, just because of the genetics and all the mutants that are very widely available, and because Salk has a lot of support for studying Arabidopsis. So what we did is just to first test our theory on them. So again, these are scans of Arabidopsis. We did the same exact analysis that I described on these. So we took the base, we took all the flowers, saliques, rosettes as kind of these suburb points. And you can see this Pareto diagram here. Again, this plant is very close to this curve, and all these other architectures are pretty farther away. So this is another species that seems to work by this idea. And then what we did was we collected a bunch of mutants. So here's a wild type Columbia background. Here's the MAX4 mutant, the BRC1-2 double knockout. So we took about 11 different mutants. I'm just showing 4 of them here. And again, the idea was to see, is there anything that's systematically changing on how these plants are moving along this curve. And so what we found, so I'll just show a schematic here, but so here are the wild type plants, and they seem to be clustering at some location on this curve. And then we looked at the PIN 347 mutant, and all of them systematically moved up closer to this part here. Okay, and so what this is telling us is that these genes are somehow, involved in making this trade-off. And if you did want to breed a plant that is more, functioning on this objective, these could be some genes of interest to look at. And then we looked at some other mutants, and we saw this BRC1-2 push the plant a little bit further to the right along the curve. The D14 pushed it even further. The CRY1-2 pushed it even further. So, these are just a few examples of how just modifying a very few number of genes can actually start to help you breed particular features of interest in these plants. So this was, you know, just looking at this, another thing that was very striking to us was that all the genotypes or all the architectures from one particular genotype tended to cluster together in a region of space along this curve. And this was a little bit surprising to us because just looking at these plants, visually they were very diverse. Like, it was not at all obvious to me that all these plants were coming from the same genotype. And so we thought, you know, could this be a nice feature to actually robustly describe a particular genotype based on this kind of trade-off that phenotype is using? Okay, and so the problem here again is that, just looking visually, there's a lot of diversity in these architectures, even replicates that we were growing with the same genetic background, in the same soil batch, in the same environment, scanned at the exact same time. And some of this is natural and stochastic, but the question is, you know, despite these changes, are there features maybe at a higher level that nonetheless remain consistent in this plant? Okay, and so the way that we got at this was, for all of these 11 mutants that I just described, we grew 5 replicates, at least 5, so about 5 to 10 replicates of each one of these mutants. And then we computed this coefficient of variation for many different features that people commonly look at in terms of architecture. So the coefficient of variation is literally, you just take the mean value of the feature, you divide by the standard deviation, and the lower the coefficient of variation, the less variability there is in that feature. Okay, and the idea is to find features that, at least for the same genotype, have a very low coefficient of variation, and that would be a robust descriptor of that genotype. Okay, and so here's what we did. So on the x-axis here are the 11 mutants and the wild type. On the y-axis here is the coefficient of variation for all the architectures in that genotype. And so this black thing here is our Pareto trade-off feature, telling you how it's balancing between the two objectives. And the other ones here are other features. For example, how many leaves does it have? How many branch points does it have? What is the convex hull volume, and other features? And so what we see across all of these mutants is that the coefficient of variation for the Pareto trade-off feature was the lowest. Okay, and again, what that means is that, however, even if the plant, even though one replicate might have 5 more leaves or 7 more branch points, somehow at a higher level, it's making a very similar trade-off despite this kind of local stochasticity that's there. And so we thought this was actually quite interesting, because it suggested a type of homeostasis that's happening in these plants, that, you know, local changes in the environment are certainly changing the structure, but there's still a feature that's very conserved. So we thought that was cool. And we actually wanted to test this over time as well. So this is an individual Columbia wild type plant. Over 4 days of scanning, I think we did about 6. I'm only showing 4 here. And what was also interesting was that even plants over, you know, relatively short periods maintained a very consistent Pareto trade-off feature. So what this means is that as the individual plant is growing and deciding where to put the next leaf, it seems to be constrained to make a very similar kind of trade-off between these two objectives. And you know, we're still trying to understand what this means and how we can use this for doing more predictive phenomics, but I think this is an interesting place to start. Okay, so the last thing I want to do is talk about whether this idea of Pareto optimality might apply to other branching structures more broadly in biology. And so I was working on this project with some plant biologists, but my lab is right next to some neuroscientists. And I was telling them about what they were doing, and they told me that, oh, you know, actually neuroscientists have been thinking about this type of problem as well. And so let me just tell you what the analogy there is. So for a neuron, instead of downtown, instead of the base of the plant, you actually have the soma, so the cell body of a neuron. And instead of the suburbs, what you have are synapses. Okay, and so what does it mean for an arbor to minimize travel distance? Well, that's Ramon and Cajal's law of conduction time. And what that means is that an action potential is generated here in the soma, and that action potential needs to reach downstream synaptic partners. through synapses, and the amount of material that this action potential has to travel is a measure of how delayed that signal propagation is going to be. So so Cajal proposed that however these complex dendrites and axons are designed, you might want to try and minimize the amount of travel distance that these potentials have to go down. And then the other objective of minimizing total length is also a well-known idea in neuroscience, which is literally to minimize the amount of wire that this neuron needs to make its connectivity. And again, we have billions, trillions of synapses, billions of neurons in our very compact brain. And so if you can find a way to make these connections using the minimal amount of wire, that could be an advantage to the brain. So again, we thought as another evolved process, how well do neural arbors kind of resolve this trade-off? And fortunately, we didn't have to do experiments here because there's this really beautiful database called Neuromorpho, where people literally trace out axons and dendrite arbors and deposit them in this database. And so we were able to collect 14,000 neurons across many different species. So here's an example of a pyramidal cell in the rat neocortex, and every region, every kind of cell type that you can imagine. And so I'll just give you one example that's showing that even neurons are following this idea. So this is a mouse somatosensory cortex neuron. So that's the cell body, and that's kind of its arborization. And we did the same type of analysis, and that neuron is also lying very close to the Pareto front and much closer than kind of these other things. And so this is all 14,000 neurons on the x-axis. On the y-axis is, again, that distance to the Pareto front, so lower is better. And so you can see, other than a few neurons out here, pretty much all of them lie closer to this to the front than the other system. So it's it was actually somewhat pleasing for us to see that these two systems have kind of evolved ways to solve this very basic engineering but also biological problem. So lastly, what I want to tell you is that we also have all our data, all the plant data online and freely available for the community. So Adam has scanned over 700 plants in the last couple of years. We have about 150 million cloud points representing our 4 species: Arabidopsis, tomato, tobacco, and sorghum, and all the mutants. And you can go in and visualize these things and do some basic analysis on this website as well. And so if there's other questions that could be interesting with this data, please let me know, and very happy to work with you. So just to conclude, I told you 4 things today. First is that at least these 4 species that we've studied have evolved mechanisms to generate these Pareto optimal arbors. We showed that we can modify just a few genes and change how this arbor is prioritizing different objectives. We gave some evidence that this kind of Pareto feature might be a more robust descriptor of a genotype than some of the other common measures that people use. And I think, from what I know, that this might be the first property that we showed is shared between plant branching patterns and neuron branching patterns, despite the fact that they diverged, many hundreds of millions of years ago. So thank you for having me, and to the team. So this is Adam, who did all the plant scanning. Ulas and Joanne. Ulas was a postdoc in Joanne Corey's lab, who bared with me as I was trying to learn basic plant biology over the last few years. Arjun is my student, who's been working on the neuron analysis. Adeline and Martin in Cambridge, who've been helping us with the mutant analysis and the funding sources. So thanks a lot.
Awesome. Thank you very much. Does anyone have any questions? And same for those online. We can read the chats for questions to you too. Any questions?
Oh, sure. How long does it take Arabidopsis? About 10 minutes. 10 minutes? Yeah, so what he usually does, so it depends. If you just want a rough scan, you could do it in less, but if you really want to try to get underneath the leaf so you can actually calculate leaf thickness and those kind of things, it'll take a little bit longer, but yeah, on the order of 10 minutes, I would say. Okay, so it'll take longer for yeah, that'll be about 20 minutes is what Adam usually took. Yeah. So that's our there are systems now that do kind of like this type of scanning. They automated, they just like kind of go in a dome and scan. Great for throughput, but now if you really, then you're kind of messed up by occlusion. You can't really get out of that. And so we figured let's just get higher quality and deal with use better data than more data.
So I can't imagine that there aren't other trade-offs that would move the architecture off the Pareto front.
Yeah.
So have you tried to simulate other architectures that would indeed, move it off the Pareto front?
Yeah.
I mean, I can think of shoot branching, mutants, and etiolation characteristics that would seem to move it off the Pareto front. And have you just picked the right set of mutants and the right set of conditions to maintain that?
Yeah, so it's a great point. We did see a few instances where the plants did move off the Pareto front. So one was in tobacco highlight plants. So for some reason, maybe some other objective kicks in, but these plants, I mean, they're not like super far off, but they are noticeably further away from the front. And so what does that mean? That could mean that maybe they're just not performing as well in that condition, or as you suggested, maybe there's a third or fourth objective that kicks in that we're not capturing here that becomes more important in that condition to the plant. And I'm sorry? Oh, Lola, yeah. I mean, so we haven't done that one, but that's so that's one case where we saw. The other interesting case we saw was in some of the mutants that I didn't talk about, the plant fell down. Like, so the inflorescence was just not strong enough and it falls down and then it tries to get back up, and then we analyzed them. It was not easy because they were like falling down, but we scanned them and they were also noticeably off the front. So maybe when they fall down, something else kicks in that's saying, okay, I need to deal with gravity and structural stability or other things, and that can also push them off the front. So it's definitely not like, there are places where the theory breaks down, which is good because sometimes then you wonder, what is actually going on here. So, but yeah. So, thank you.
For the figure you showed that was showing how number of neurons in the systems you looked at related to how close they were to the Pareto front.
Yes.
So to the far right, as you get to more and more neurons,
yes,
there was a spike there.
Yeah, some of them. Yeah. So okay. So these are either they're just not Pareto optimal for whatever reason, and Arjun is actually looking into now. Is there something systematic, like maybe they're all from the fly mushroom body, and for whatever reason the fly mushroom body isn't abiding by this? But yeah, you're absolutely right. There are some exceptions here as well, but I can't tell you why they are biologically.
So it's not like a particular set of species, or
I don't know yet.
Okay.
Yeah, so this is literally, I got this 3 days ago.
Okay.
So we're looking at it now. But yeah, it's actually, this is probably the most interesting part of this plot. Like, where is it not working? And yeah. Cool.
Thank you.
All right. Do we have any last questions for today?
Stems,
yes, yeah.
Some of the other.
So we haven't in this case, but there are clear cases where we see thickening. Like, for example, in highlight, we see almost a twofold thickness change in the stem, and so we could incorporate those in our graph in terms of weighted edges and a weighted measure of length and travel distance. We haven't done that yet, and the reason we haven't is because we're trying to automate the capturing the actual weight, the actual thickness of the branch from our 3D scans, and that's been a little bit more problematic than we were hoping for. But we are going to try to get to that point where we can not only do the thickness of the branch, but also the size of the leaf, because that's also a function of how many sugars or something might have been generated by that leaf versus another leaf. So, yeah. No, thank you.
Awesome. Last question? All right, let's thank Saket once again for his amazing presentation.